There we go. Hi, everyone. Good morning. Again, yeah, sorry I'm a bit under the weather, and I apologize in advance for coughing if I do. Okay, so as Robbie said, I've been working for GrainGenes on and off for about 23 years, and this is our current team. Tanner Shen is the director. Jerry Lazo has been there the whole time. He's a geneticist. Me, Eric Yao develops and programs our JBrowse, which I'll be working with a lot today to show you. And Steve Michael is our IT guy, keeps us safe. Okay. So hopefully most of you are familiar with GrainGenes. It's been around for nearly 30 years now. Can be found at wheat.pwsda.gov. It's a very inelegant URL, but there it is. We have a liaison committee made up of experts in the field. And for many years now, they've been trying to tell us that we need to move from a genetic to a genomic context. Okay, so back at the at our homepage again, there's a at the top there's a group of quick links. Okay, and of these, one is the Genome Browsers. So when you click on that, the very top pane of the browser selection menu is basically the most popular one for each of the crops we use. And if you scroll down a little bit on that page, you'll come to barley. And when you click on the pan genome, it expands to all these different cultivars where we have browsers. So at its very core, a genome browser is really just the sequence of the DNA. If you kind of can think about this top line here, these are base pairs. So this is Barley chromosome 1H from one telomere to the other, just laid down on its side. And again, at the very core, it's just the nucleotides, and we call it the pseudomolecule track. So here, these are the colored blocks of the nucleotides. The gray boxes, depending on the frame, are amino acids, and these little red guys are the stop codons. Okay. Something's loud in the background there. Oh, is that Jerry? Anyway, so here I've expanded out, so now that's 50 kb. Then the last slide, 50. This is now 50,000 base pairs. You can no longer see the sequence anymore, but you can start to see the genes and gene models, which what those are. Okay, and if you expand out even more, you can't see individual genes anymore, but you do get a graph of the feature density. I'm not sure what's going on there, but there it is. Okay. We get this question all the time, is how can I save sequence from or the sequence from a region in the genome? And this little video will show you. Excuse me. So you click on that bar, scroll it over, and it will zoom to the region. And then when you mouse over the pseudomolecule track, you'll see a little downward triangle. Hit Save Track Data, and then from there, excuse me, you can select a FASTA file that will download automatically for you. Okay, so I showed you that we have the nucleotides, we have genes. What else can we do to put on the browser to help us understand it? There's lots of marker types, but can we overlay QTLs and regions of the genome that provide traits? We might not know what genes are involved yet, but we know they're involved in traits, and we use those regions of chromosomes in breeding. So now that we have fairly inexpensive sequencing and other things like that, we can start to look at a lot more data at once. So a new technique that people have been using now for, I don't know, 10, 15 years is called genome-wide association studies, where you take a large diverse population, you measure the traits, and then you analyze the variations for DNA in the DNA to look for correlation. And those variations are usually SNPs, single nucleotide polymorphisms. Okay. So this is out of Wikipedia. This is the human genome, the 22 autosomes. And these are genes that are known to contribute to kidney stone disease. So basically, each of these little dots are one marker in human that they've measured, and the higher up on this axis that the correlation is, the more likely these are to correlate to the disease you're looking at. Also in barley, we can do the same thing. So these are the 7 barley chromosomes. Each of these are markers that we know, and in this case, they looked at grain yield. So we might not know what they are, but we know this SNP is involved in grain yield. Okay, so this isn't barley, this is Triticum turgidum, but the McAferry's group years ago gave us these data where this grad student went for, sorry, excuse me, about a year probably, curated all the QTL for Durham out of the literature and overlaid those on a track on the Genome Browser. So that kind of gave me a bit of an idea to start doing that same thing for the other crops that we look at. So this is the GrainGenes. Let me get rid of this. Maybe there we go. The GrainGenes update panel that you can see on our homepage, and we just made this announcement a while ago. So I started with barley because we hadn't done a lot for barley in the last few years. Went through Google Scholar to look for GWAS papers. And what I noticed was that a lot of NetBlotch QTL papers were out. So started there, worked on some other diseases, agronomic traits, and a malt quality paper. So I did all of these before we made the announcement. And this is the result of the NetBlotch on 6H. So you can kind of see there's a lot of QTL down around this region, a few of them up here. And I just wanted to show you, if you mouse over any of these, it will show you the name of the QTL. And another little movie here. Okay, so you can go, let's start. Okay, so you click on that, and it'll bring up a report page. So all the SNPs, these are all links back into GrainGenes. Pardon me. And then the source report or the source link will take you to the reference. And that's really the one place in GrainGenes where all the QTL from that study will be shown. Sorry, you guys. Pardon me. Okay. So, and then if you click on one of these, here's, I just picked a random one. Click on that. That little glyph next to the QTL is a link that'll take you back to its position on the browser. Okay. And okay, I am really sorry. I'm going to have to finish this in a minute. My sincere apologies. Is it okay if I just continue after? I need a second. You're muted, Robbie. Sorry. Okay, here we go. I got it. Okay. Sorry.
Robbie, we can't hear you.
You're muted, Robbie.
Okay,
I've got people coming from every angle telling me that I'm muted. So,
no, it's okay.
I was just saying that, you know, I think everybody can hear that you're struggling just a little bit. So if you want to finish just now, then please do. And I'm just wanted to ask Craig if he was all right and ready to take over.
Okay, thank you.
So anyway, Craig, over to you. Vic, go to your bed.
It's tough when it's like that. So yes, so I'm going to talk to you a little bit and describe the Barley gene and transcript abundance data set that we've been setting up. So the main aim of this is to kind of give researchers access to Barley gene expression information. So my presentation will have 2 parts to it. One part I'll just kind of go through some slides and introduce you to EoRNA and what it is and what sort of data it has and what you can do with it. And then the second part is the practical part, which Robbie was asking us to set up. So I'm going to try and do this live. So if you do have a FASTA file, I'll kind of unshare and I'll grab that FASTA file and we can put it into EoRNA and see how it goes. If not, I do have one example I can show you as well to kind of show you how quickly and easily you can generate information and data. So I'll just start from there. Okay. Okay, so we can now measure the abundances of transcript expression reasonably easily, and it's really down to this revolution in sequencing, the ability to sequence large amounts of data and generate transcriptome information. So we do this by RNA-seq, and it generates millions of these short read sequences, which can be mapped to genomes or reference transcript data sets. And importantly, these can be counted. So what we use for quantifying that is a couple of pieces of software. They're called Salmon and Callisto. And what these do is use fast allocation of these sequence k-mers, and they allocate these to reference transcripts. And the real key thing about these two bits of software is that they're really fast. So you generate fast transcript quantification of your data. But to do that, you require an accurate and comprehensive transcriptome. So a lot of our work in plants has been built up from a couple of people here at Hutton. So Runjuan Zhang and Wenbing Guo had set an RTD, a reference transcript data set, up in Arabidopsis. And we wanted to follow that in Barley so that we could quantify RNA-seq data very quickly as well. So our first go at that is what we call BART version 1. It's Barley Reference Transcripts, is what it means. And that was done by Paula Flores and Misha Bayer here at Hutton, and got that set up in 2019. And so then we can generate values for RNA-seq data sets. And the output for that we get in transcripts per million. So this is a commonly used normalization method, and that considers library size, the number of reads, and effective length of the transcripts. So that's how we're generating the numbers. So we wanted to look at this across a range of different RNA-seq experiments. So we accessed public archive and also in-house Illumina short-read RNA-seq expression data. So this came from a broad range of different barley cultivars, different populations, different stages, organs, and also biotic and abiotic stress conditions. So it gave us a really broad range of different tissues and RNA-seq data from those different conditions as well. So the total for this kind of first version that we've done was 22 different RNA-seq experiments that we downloaded, which covered 843 different sequence samples. And these were all quantified using Salmon. And then what we do is take those TPM values and very simply, we just plot them. So we do not consider the batch differences between samples. We do not consider differences among the experimental studies. We're not working out statistical significances, and we're not looking at scaling things like log2 scales. We looked at all those and we felt it was the best thing we could do is just give the raw TPM values. And then what you can do is then download that information, and you can then do all the different statistical significance that you want to do. So what you end up with these plots is a really intuitive, rapid visual assessment of transcript expression levels of barley genes. And we found these extremely valuable in comparing different sample sets and what each of the genes are doing in terms of expression levels. So this is what it kind of looks like. These are the sort of outputs that we get. They're gene expression plots. So this is an example of one gene only. And along the bottom, you have the samples. So across that bottom is the 843 RNA-seq samples. And then up the y-axis, you can see the TPM values. I'm just going to point our options. So yeah, so along the bottom here are all the samples. And then up the side here is the TPM values for the expression levels. So you can see very clearly a number of different things is that you're getting very different levels of expression numbers in the different tissues across all the samples. And you can go and look in these individually if you wish to. The other thing that we have is a number of different transcripts. So within every gene, there's a number of alternative transcripts that often get made through things like alternative splicing. And I'll give you other examples like cultivar differences as well. I'll show you some examples of that in a little while. But you can then see this, and this is what's quite unique about this expression data set, is you can quantify individual transcripts, and that's something that you can't do in some of the other formats that are very similar to this. For every sample that we have a little metadata tab. So you can see here that you've basically got the RNA-seq sample set that was used. It shows the cultivar that you've got, and also gives you the tissue, what replicate, and then what sort of condition that was there. So you can slide along these 843 samples, and you can look at see where all that information is coming from. Okay. So one thing that really jumps out when you're looking at this data is the stuff that is really highly tissue or condition specific. So I've given you some examples, 3 examples there. One on the left here is from developing grain. So you've got a gene that's quite uniquely expressed here, and you can see that in all the kind of tissues that it's coming up, it's kind of related to developing grain and also within the spike. So this is quite a nice way of identifying uniquely expressed genes. This is an example that's to an abiotic stress, so low temperature. Again, we've got 2 experiments within the sample set, and you can see very specific levels of expression that's going on there. And then we also have examples of biotic stresses as well. So this is to fusarium, and you see the really high levels of expression for this gene here, this PR gene. So the thing to remember about all these things is that this is all done within the context of 22 different RNA-seq experiments. So you can identify genes very easily, and you can see the connection between the expression levels, and then give you some real confidence that this is a gene that is really quite important in that response or within that tissue. This is another example here where you've got an induction of a mutation. So this is a gamma radiated mutation in Morex sample set, and it was selected for its susceptibility to spot blotch. So again, here you can see the effect of that mutation on the expression of a target gene. And of course, there's also possible transacting effects downstream of that. So these were 6 different experiments, and you can see the expression levels have gone in the mutant compared to the wild type Norex. So to get to the unique feature of this data set is the transcript variants. So firstly, you can identify cultivar specific transcript variants. So this reference transcript data set was built up against the cultivar Morex. So we kind of describe it as a Morex reference transcript data set, but we did include a large number of different samples from different cultivars, and some of these come through. So this 4 base pair insertion indel is there, and it generates a unique set of transcripts. And you can see those. So you've got the quantification that you can see the difference here between the cultivar Clipper and cultivar Sahara. And you can see that all the way through the different sample sets. So, and the difference here is this 4 base pair indel that you can identify. So it's a really nice way of looking at the effect on transcripts of from different cultivars. And then the other way that you can generate alternative transcripts is through what's called splicing, alternative splicing. And you can pick out these different transcripts very easily. So in this case, we've got this gene that's got 10 different alternative transcripts. The majority are in this green type, number 3, transcript 3, or you also find this blue one, which is here. So the difference between the two is these 2 transcripts is an intron retention, and that can affect the function of the protein. So this is for a developmental step. This is also one that's related to cold stress, and so you can pick out this variation very easily. This is a set of samples that are showing at 24 hours, you get at 4 degrees C, you get this switch in the alternatively spliced product, and you find this transcript, this purple transcript, which has another intron retained within it. So it's really nice way in which you can pick out these different alternatively spliced transcripts. So I'm just coming to the end of my first part. So where are we going with this? So We've just had a new version of the Barley reference transcript RTD published. So we call this BART2. So this is based on a different cultivar, Barca, and it has improved accuracy, particularly at the 5' and 3' ends. And this is due to the isoseq data, which allows you to define these 5' and 3' ends much more accurately than we did before. And what Misha has been doing is automating the downloading of new RNA-seq experiments. So we are at this point in time looking at around about 5,000 samples covering about 122 studies. So EoRNA2 is on its way. The challenge that we have is how we display over 5,000 samples. And you can see that picture at the bottom here. This is kind of showing all 5,000 samples for this one particular gene. So that's kind of the challenge that we're looking at the moment, and we're working away on that. The other thing we'd really like to do is start incorporating the information, the sequence information, into existing visualization tools. So the example here is the bar expression browser. So generate some nice pictures of tissues and then add in kind of levels of expression within those tissues. So that would be a really nice thing to do, but that's the sort of things we're looking to do in the future. Okay, so what I would really like to do is then go and explore to go and explore the data for your own favorite genes. So this is what you can do. So what Linda has done is put together a website that allows you to very easily do this. So this is the front page of it, and I'll go to the front page of it live in just a little while. But it's just the keys into that expression data, and you can do that primarily. The way I primarily do it is through BLAST searches. So you can go and set BLAST up, and you can then identify the gene or the best match to gene or multiple genes, and you can then click in and you can find out the expression levels that are going on with that. If you already know a HORVU gene, you can go in via a HORVU name. So this can be added in here, and you can go and search for that specific gene. If you are aware of the BART transcripts, you can go in immediately using the BART reference transcript as well. So these are the sort of numbers of genes that we're dealing with here and the number of transcripts that we're working with. Okay, so that's the front page, and what I'll do is I'll go through that front page and let people have a look at it, see what the sort of information that's there in a little while. So I just at this stage want to acknowledge people. So Linda Milne has been developing that, developing the website. Misha is the one that's been involved in all the way through in developing the reference transcript data sets for Barley. And also he's been helping us download that sequence information that Linda puts into the website. And also I mentioned to Klaus, who's our statistician, who's kept us right with the statistics. And we've also had the help from Ranjuan and Wenbin, who were working with John Brown in developing these reference transcript data sets, and originally in Arabidopsis. And then Robbie, he kind of gives us cups of tea, keeps us happy. So how do we access EoRNA? So if you go into your Genome browser and just type in EoRNA database, if you go into EoRNA and just type in EoRNA database, you will then go to this site here, and that gives you the front page, and you can go from there. So I thought I would just kind of do one thing to kind of highlight how quickly and easily you can generate information and data and set up your stories, set up some projects or whatever that you're interested in. So I had nothing to do with any of this until yesterday. I had a look at this, and what I got interested in was something called the Lordicule and Lordicule expansion. So the Lordicules are involved in the closed flowering in barley, and where you have a Lordicule to generate the flower, it expands and opens up the flower. And in the majority of our elite lines in barley, the flower is closed, which allows for self-fertilization. And there was a paper came out in 2009 that described the mechanism by which that happened. So this was a gene called CLY1, and it described this gene as being expressed, and its regulation was through cleavage by a microRNA, microRNA 172. And you can see in the figure on the right here, so that the difference between those that had the closed flower and types that were open flower, you have this mutation here which affects the microRNA targeting site. So this is one nice mechanism by which you generate these highly reduced lordicules. So you can go and find that BART gene sequence if you like. The other thing to find out is that there's a patent associated with this information as well. So if you look at the expression of it within the Barley reference transcript data set, you can see that there's a lot of expression everywhere across all the samples. This is a data set here that's from microspore, and you can see that there's no expression at all. And this is Lordicule information up here, which shows that it does have good levels of expression for this gene. So as it's related to a patent, it'd be actually quite nice to have some alternative gene targets. So you can go and look in rice. So rice has a number of mutants that are related to closed flower. And one of the main mutants that's described and is commonly described and commonly used is a gene called Superwoman1. And this encodes for a rice class beta MADS box transcription factor. And it's specifically expressed in lodicules and stamens. So the mutation is a substitution, and what happens is you get a reduction in the interaction with two other MADS box transcription factors, which leads to the closed flower phenotype. So what I'm going to do is I'm going to look for a MADS16 in barley. So I'm just going to come out now. So stop sharing. So I just need to come out of, here we go. Just get something set up and then I'll start sharing again. Okay. Okay. I can't get out of my, there we go. Okay, so what I've done is just to type in MADS16 gene into a genome browser, and you get these really nice pages that describe these kind of genes in rice. So if I click on that, I get this list here. I'm going to go to this site here to pick up some sequence. So it generates this information. I can download the sequence. And what I tend to do is go from with peptide sequences. So I can just quickly copy that. And then I can go to BART. So if I type in, if I just type in EoRNA database, I can then find EoRNA Barley Gene and Transcript Abundance database, and we can go straight to this. That's not what I wanted to do. I want to go here. Sorry. So this takes you into the website, the EoRNA data website. And there's kind of basic information up here about where all the information came from and what we've been doing with it. But the way I tend to go in is via BLAST. I'm going to show you the annotation search first. It just kind of shows you can pipe in various keywords that you want to, and it'll generate a list of genes. If you want to translate a list, now this is something that's ongoing at the moment, and I wouldn't recommend using it just now, but what you can do is you can type various versions of a gene in here, and it will then identify the BART2 transcript. So it's maybe not overly helpful at the moment. And then there's also, we've got a tool that allows us to look at Go enrichment as well. So if you generate a list of genes that are of interest, there are two ways to look at or identify Go tags. One is to go in here and you can open up a Excel spreadsheet, which has a lookup feature in it. You just basically add in your list of BART transcripts, and it will then automatically link these transcripts to Go tags. But you'll be able to see this already in the pages that I'm going to show you. And then there's another way of doing it is to put this in here. And you need for this, you need a GMT file, and I'll show you where to get that in a little while. So you need this GMT file to kind of hook it up to the BART transcripts. Basically put in your links, and it'll generate these kind of really nice images which show all the different Go tags that you can get. Okay, so I'm going to go BLAST. I'm going to put in my sequence, this MAD16 sequence. I need to select the sequence type, so it's protein. I'm going to take the default here, and then I go to select the BLAST database. So you can do reference transcript data set for BART, which is the one I'm going to use, or you can go via HORVU if that's the one you wanted to do, or you've got access to also the MOREX genome assembly. But I'm just going to go straight to BART. And what that then generates is a whole list of genes that have some sort of identity with this MAD16 gene. Obviously, the one at the top is the one that everybody clicks and looks to, but you should also be aware of what these other transcripts, transcripts are like and what they may be showing you. For me, this is really nice because it's kind of covering the whole length of the sequence that's there. There's a good match. There's a little bit of variation in the match down here. If you want to see the full BLAST results, you can go click there and you can generate that information, and you can go and pull that out if you like. So I'll go back here. So now I'm going to go into where the actual information is. So I've just clicked here, and it's a way to open up. So because these are individual transcripts, it takes me to a page that's got the individual transcript information, which is just the sequence information. If you then click on View Gene, you can go to that, and then this is where you'll get the information that you want. So here we have all the 843 samples that I've shown you before. This is the level of transcription that's going on, and this is the expression values that we see for that specific gene. And you can see there's like a really high band, a really high peak here in this one. So the nice thing with these sort of graphs is you can grab them and you can go in really quite close and look at the information. So you can look at the tag, and that tag is now telling me this is Lordicule specific. It's really high levels of expression in the Lordicule. You're also seeing a little bit of level of expression in the inflorescences as well, and a really small amount in the Pallia and Rachis. So I can then go back to so this is basically showing me that I've got a very Lordicule specific expression of that gene. So it becomes really quite an interesting gene. There are 3 transcripts described for that, and you can go to the individual sequences for all these transcripts just by clicking on these. Obviously, from looking at this, the main transcript is transcript 1. So this is the gene model for transcript 1, and just shows you the exon structure and the introns in between. If you want to find that gene as a HORVU number, you can click on there and go to that. The information here is really quite important. It kind of lets you see the sort of level of overlap that you have with the HORVU gene sequence. So in this case, it's at 98.1%, which is pretty good. And then the information here also shows homology to the different plants, so largely rice, Arabidopsis, and Brachydipodium, and it shows the identity of those genes that you can go on, and you can then click on those to go and visit that site if you wish to. As I mentioned earlier, you've got your Go tags. So these were generated by Misha using Panzer, and you've got a whole list here of your Go tags associated with this gene that we've just pulled out. And then kind of similar to what Vic was saying, this is not JBrowse. This is kind of the earlier version, GBrowse. So you can click on the GBrowse and go to that. It kind of shows you the gene models of the transcripts that you've got for BART. It also shows you the transcripts that you have in HORVU. And I'm not sure, I'm going to just click on it just to see. It also usually shows genes, so not in this case here, but it usually shows the position of the 50K Barley SNP. So you would see that there may be like a SNP from the 50K chip that's associated with the gene, but it's not showing you anything here at this time. And a bit like the JBrowse, you can click on that and you can go and get hold of genomic sequence and also transcript sequence. Okay, just going to go back. So that's the information. I then, because I got kind of interested in this, I kind of went back and I did also looked at the MADS2 and MADS4 and pulled those out, and lo and behold, they are also Lordicule specific. So basically from yesterday, I had no idea about any of this, and I've already identified a gene that's very specifically expressed in Lordicule, which is really important in the flower opening and flower closing, keeping the flower closed. Okay, so I'm going to stop. Well, there is one other thing I can say is you can then take this away and you can go to something like Cranachan and identify some SNPs associated with those genes across variation over a large number of different accessions, land races, and wild types. Okay, I'm going to stop there, and I'll happy to get questions.
Craig, that was great. I think you've stimulated quite a lot of discussion and ideas in the chat boxes. Can I maybe suggest we do something first of all that we said we would do, and that is, can you go into the chat box, Craig?
Yeah. Yes.
There's two sequences in there that have been pasted in by members of the audience.
Okay.
Could you go through, could you give us a demonstration with these unknown sequences? And
Yes.
then we'll ask the people that supplied them to let us know why they want to find out about them.
Okay. Maybe they're private.
No, they've even got their names on them.
Okay, so this is I'm going to pick up this heat shock protein 90 from Luke first of all. So I'm going to start sharing again. So I've just copied it at the moment. So can you see that now?
Yeah, we're back on the browser.
Okay, so this is that heat shock sequence, and it's now nucleotide, and I'm going to do Barley reference transcripts. So it's coming out with a really strong case. There's actually another one that looks very good as well. So, I mean, I would be looking at both those. So there's two genes here, it's like 07178 and 37203. The other ones, these are kind of alternative transcripts from that same gene. And then there's some sort of similarity. Now, when you see something like this, it's usually some sort of domain that's conserved, you know, like a DNA binding domain or, you know, something like that. That's conserved, so it's maybe not as related to the gene that you want or of interest. So again, I'll just go to the top one to look at it, and it will start opening up. Okay, so this is another really nice, quite specific case as well. So this is kind of in hull-less barley grain down here, this sort of level, but this is obviously the one bit that's kind of really interesting. So where is that? So that's coming from heat stress root tissue. So basically, I think from the name, the title, it was telling me it was a heat shock protein, and I've just gone into our sample set, and the really top level expression is tissue from root, which has been exposed to 35.5 degrees Celsius. So it really kind of supports the idea that this gene is actually quite important in heat stresses.
Okay, so now I've got a question for Luke. Why were you interested in the heat shock 90 protein? Craig, maybe you can grab the other
That's what I'm doing.
thing that's there. If you're talking, Luke, you're muted.
Hey, hello, can you get me? Yeah, it is a heat shock protein that Miriam pulled out as part of the background bioinformatics for the shuffle meiosis project. It wasn't particularly Maltic specific, but it's interesting to know where it's actually being expressed.
So if I never knew from the title, I mean, I would have got that sequence and gone, oh, look, it's like really highly expressed within this heat stress tissue. I then could have gone to the list of like tear and rice genes, and I could then click on those, and I could go and probably get some information on what that gene was.
Okay, Craig, quickly, we'll get one more, and then we can go into the discussion or the advice or what
Yeah.
suggestions.
So this one I don't know anything about. See what happens with this. Oh, it's unique. It's found nothing else other than this one sequence, but it's very, very nicely matching. So this has got quite a lot of general expression across all the tissues. So it's described as a zinc finger protein with 78% match with rice, which I could then go and find out a little bit more about. But it's generally found across all the different tissues. If you were working with a specific tissue that you were really interested in, because you can see that there is some variation between different samples, and you could maybe go in a little bit closer. So let's just go in here for a bit of an example. So you can see here that there's some sort of internode that seems to be slightly higher than the other samples that are kind of all around about this kind of 20 level. And then this one's going up 25, 35, 40. So it's 3 replicates. So you've got replicate 1, replicate 2, and replicate 3. So, and it's from these developing tillers at the 6-leaf stage in the 3rd internode in more eggs.
So, Dominica, do you want to tell us something about it? You might need to unmute.
Can you hear me? Yeah.
Yeah.
Yeah, so it's the ST1 gene, and we know it's GRAS specific, and it's probably E3 ubiquitin ligase. So it is quite well expressed all over different tissues. So yeah, I think this is what I would expect to see.
Okay.
Good. So there you kind of go. You've got something that's kind of supporting what your thoughts are, and you've got information there to support it. So if you were writing a grant or something, you expect this to be expressed everywhere, then that information is there to kind of support that.
Yeah. So is
Okay.
it possible to check if it's specifically expressed in a particular tissue?
Yes. Yeah. So you can go and just identify this. You can go along the different experiments. I'm trying to think where the table is. There is a table somewhere. It's definitely in the paper. Where it lists all the different experiments with the different tissues and samples. So you can identify the experiment, and then you can go in and you can identify that in the sample set across the bottom.
Okay. Okay, great. Thank you.
Okay.
So, Craig, there's a lot of questions actually, or maybe some advice in the chat box.
All right.
So you asked the question in your seminar, and I asked it to everybody in the audience about whether or not they had any ideas about how to display more than 5,000 transcript datasets.
Yeah.
So Neil suggested maybe a cluster analysis first to reduce complexity.
Yeah.
Is that?
Yes. So we can use the metadata as one thing we've been thinking about, in which you can get the whole data set if you like, but you can then kind of click on a box that has a list of all the developmental stages, or kind of linked to the metadata, and you can then just pull out a specific cultivar, for example, or you could pull out things. So that would then reduce things a little bit. Linda and I have also kind of thought about moving it from kind of going horizontal to vertical and going down, but you still have the same problem. You're kind of looking at different things from kind of top to bottom. I don't know if there's kind of a way in which we can maybe, we talk about these things and we're kind of trying to figure out what the best way is, but one other way is to kind of list them by the kind of top TPM value and go all the way down, but then you start splitting up all the replicates, that then kind of makes it a little bit more difficult. One thing I should have shown you and didn't was at the bottom of these graphs, there's a little bit you can click, and you can download all the TPM values as a text file, which then you can do all your stats all you like. So I actually did have a file set up to show that bit.
So David Horner suggested, or maybe you could just order them by the delta within experiments.
Within sorry, I'm not seeing that one. Order them by the what?
329, the delta. Well, David can explain what he means by that if he wants.
Yeah.
Hang on.
You mean the difference?
Yeah, do you want to tell us, David? Just the difference.
What? No, I just thought if you took the. I mean,
We can't hear you very well, David. Sorry.
Sorry. Can you hear me now?
Yeah, perfect.
Okay, cool. I just thought if you just like, I mean, I know you're staying with simple TPM values and stuff, but if you just ranked experiments, not splitting replicates, but take a mean for replicates and take a full change delta and then rank the experiments in the order that you show them. I mean, it's very simple, very crude. But then you see as the first things on the list, always the things with the biggest delta of expression. It's just a quick and dirty way of ordering the samples.
Yeah.
Obviously, specifically for each gene.
Yeah, I think that's a good idea. We can have a look at that.
So Nikolai suggests that rather than blasting every time, you could probably use Ensemble Plant's gene tree data.
Yeah, so I don't know that at all. So I can certainly go and have a look at that and try using that. I've not used I've used Ensemble. I found that really valuable way of getting hold of sequence and gene information, especially some of the genes that we kind of, you know, you might get a Morex version 3 gene that we don't really know much about. We can get that sequence information, then put it into BART, try and find the BART. So I've used Ensemble quite a bit, but I haven't used it for something like that. But
Would it be useful, Craig, for you and Nikolai to have a discussion offline about that, how this could be used?
Yeah, I mean, I think it's a case of me going and having a look to start with and see what it does, and then yeah, if need be, I can get in touch with
Okay.
Nikolai.
So I think Nikolai only provided his first name, so I'm not sure what Nikolai is. Do you want to introduce yourself, Nikolai? Maybe he's gone.
It's all right.
So David again has another suggestion. He said plaza subfamilies also where direct orthologs are obscure.
Yep.
I guess that's in relation to the gene tree.
It's the same. It's basically the same as the ensemble story. It's a set of gene subfamilies. So they've done trees, and where direct orthology is not obvious, you've got groups of subfamilies, and it's just another way of doing if you don't have direct orthologs, identifying the closest and most likely in paralogs to a degree.
Okay, I'll look into that. Plaza subfamilies. Yeah.
Maybe they're called Plaza ortholog families or something like that, but there's some stuff at Plaza that's potentially useful for the same thing as Nikolai was suggesting for the ortholog identification.
Okay.
Okay, Craig. So Tanner suggested it. Would it be helpful for users if you gave links from the Horvu genes from the pages to the Morex3 genome browser to GrainGenes and vice versa?
Yes, quite simply, yes. So with BART2, Linda has basically created it. I'm going to call it simply a table that kind of associates all the different BART1, BART2 transcripts with all the different names for genes. So MLOC, JLOCs, Morex version 3, Horvus, everything that she can find, and she's associated those transcripts. So you can jump into, well, you should be able to jump in easily to find the expression data for each gene that you're interested in.
Great. And when will that become available? Any idea?
Good question. So part of the difficulty, there's a couple of difficulties. One is dealing with the metadata. So we've been having to kind of do that individually. And that's for 122 different experiments and the information within those experiments. partly because the information is quite variable at ENA. So where the nucleotide archives are and the information that you get there, you don't always get the information that consistently, and it can be quite difficult. So there's a bit of time and effort going into generating the metadata table.
Okay.
So that's taking up time. And then the time is kind of how we kind of set up the thing that we can visualize all this sequence information, this expression information.
Okay, great. So I mean, a conversation with Tanner and the GrainGenes people might be useful as well at some point. Laura asks, how do you connect to Cranachan? So maybe you could tell people what Cranachan is first of all, and then ask how you can connect to it.
Yeah, sorry,
So,
I asked because you mentioned it, right, Craig? You
yes.
said you can connect. So I was wondering if it's become public, and can you share some information?
I can show. So basically you have to open up the Cranachan website, and then you can get sequence position information for the gene of interest. So you've got a gene that's expressing in an interesting way, and then you can go to it. If I share again, I think I do have that information there.
Do you want to explain what Cranachan is to people?
Is
Craig?
Cranachan public now? Because it wasn't until not long ago.
So yes, it is. I went on, I kind of searched for a website and I went on, maybe it's still closed. Maybe I got in because I'm hunting, I don't know. But
Can you share your screen and show where it is?
Sorry, Craig, maybe I can just jump in here. So
Yep.
at the moment is still effectively just password protected and private access because it's still under development. And the data behind Cranachan is a large data set of exome capture data, which has got about 1,300 samples in it. But at the moment, the Cranachan interface is kind of, the development is on hold because the developer left a couple of years ago, and we're only just picking it back up again. So it's not really fit for public consumption yet, but it will be hopefully at some point, in the not too distant future.
Okay. So here, this is Cranachan that I was able to get in. So I'm grateful I can get into it. So I basically put in the position of the gene that I've just been describing, this MAD16 gene, and you can then select different sample sets. I'm just going to use wild barley consortium. It's a smaller set and it's quite variable as well, so you can pick out genes. And I'll maybe show you the wheelbay one as well, see how it goes. So I can click on that and extract that information. So you can then generate this information. What I do is just directly download that, save that. And then I can open up in a program called Flapjack.
So all of these programs, Cranachan and Flapjack's published and it's been used widely, but Cranachan is, this sounds like it's a Hutton specific database at the moment.
Okay.
And not everybody can access.
So this
Actually,
is
this relates also to the question from Udza. She's also asking, or he's also asking, I don't know, is there any way to identify SNPs using this database? So it's
so
kind
this
of
is what
connected.
I can show you now. So if you go to the color scheme, change that to that. So basically you then identify these different variants. My picture's in the way, so if I zoom that kind of all the way out, so you can see quite clearly there's like some really nice potential SNPs that you could pick out if you wanted to go there. I'm going to go back. So if I go back here and let's just try the wheelbay data set and extract that. And I'll download that. Michel will be so proud of me because he showed me this just last week. So this is a bigger data set. And again, I'll change that. Okay. If I come back here, so you see that this is much more, there's very few cases, but down in this here, there's a group of, so I click on it here. So you can then pick out these different SNP variants within this part of the population. So this gives you just a number. So again, you can take that number and go back. So that it's kind of hard to do it with. So if you go up the top here, so this position here, you can see in the red there's a number, that 557248665. That's the position on the genome. So you can then go back. You can then go back and you can try and identify it from there, where it is. So it's, I mean, this is exome capture data, so there's a good possibility, but not an absolute possibility that this is within a bit of exome, so a bit of coding sequence, which then you can design your caspasses and do what you want to do. Okay.
So I'm just reading down the comments now from Uzal Linton in Canada. Is there any way to identify SNPs using the database, which is what you've just shown? And if yes, how do SNPs in intron regions come up, as it was the messenger RNA that was used for sequencing?
So say that again.
I think I can probably answer it. The database that Craig just showed is based on exome capture, but the reads from exome capture go across introns very often. So you can, on occasion, identify SNPs in the 3', 5' untranslated regions and introns, although the most robust ones, we have to say, would be present in the coding sequences, because that was what was used for exome capture.
So if you saw the image, I'll go back, may as well share it, sorry. Let's just go back to this. So if you look at this here, you can see roughly where the position of these introns are, because there's little bits of sequence that have been found within the intron, and then it's been included because it's different from the others, other samples. So this kind of big white gap here is probably highly likely the position of an intron, roughly. That's what I would guess.
Yeah. When will EoRNA 2 be ready?
Yeah, that's what it's hopefully not too long.
Okay.
I wouldn't like it.
Will you plan to make that available to the community before publication?
Yes, what I would like to do is kind of make a kind of update type thing from BART 1 to BART 2. And so yes, that should become available. We'll probably do some sort of bioarchive to kind of start that. There's a few things that can be said about BART 2, and we could maybe try and do some sort of update type paper. You know, I don't know, it wouldn't be a full blast paper, but this was BART 1 was published in Nature Science Data, and I'm going to go back to them and see if they take updates, and like a few pages.
Okay. So listen, I'm conscious of the time, and maybe Vic ducking out was a good thing in the long run, because this has already taken just over an hour and we only heard 5 minutes really of GrainGenes. So what I'm going to suggest is that we have another session at some point which gives an overview of GrainGenes. Tanner, I'm sure that'll be okay. But I'm also conscious of the fact that around our community there's other resources being developed which are perhaps only used in-house in certain institutions that would be really helpful for us all to know more about. I mean, IPK, for example, I know must have software that is used in-house that they could widen out to the community, and maybe also in Canada or Australia there might be some stuff there. So I mean, I think if anybody has any more questions, please put your hand up and shout. If not, what I'd like to do again is just thank Craig and Vic, of course, for getting out of sick bed to start talking to us about EoRNA and also about some of the developments in GrainGenes. Does anybody want to say anything about whether this has been useful or not, or whether we need to have another session on these things? If you do want to say anything, please unmute. I'll give you a minute or a few seconds to unmute and say anything that you want to say.
Niels.
Yeah, I think that was a very useful seminar. And there is, for those of you who make it to Riga next week, we have a data session. The last, I think on the last day, there is a session about Barley data resources. I think you, Robbie, are chairing. Huh?
Quite possibly.
I have to give a seminar there, Craig. I would be happy to reuse some of your slides. But I think I'm planning to do less of a real a keynote lecture on that, but rather an interactive to extend on what we have started here. Because that's really something where we also really need to bring in the new generation Barley researchers, because for long-term sustainability of these resources, it's really time that we have more young people also that drive that.
Yeah. So what Craig said or talked about today, for many of us on the call, would have been quite quick. And I'm sure we can't remember everything that he said. But just to remind you that the presentation that Craig's just given will be on the International Barley Hub website. So you can go there and you can have Craig speaking and going through EoRNA at the same time as you're trying to do it yourself, just following his instructions. So I think that would be, yeah, I think that's quite valuable for these types of presentations where we can go back to them and look at them slowly. Donna's just said we'll reschedule Vic's seminar for after the summer break. We will indeed. Some nice comments from people about how useful the session was, and I would tend to agree. I think that's been really useful and really good, Craig. So thank you very much. Thank you all for turning up. And we're going to have a break now over summer because we've got holidays. We've got the International Barley Genetics Symposium coming up. We've got the Australian Barley Technical Symposium coming up, and a few other things which are just things like holidays, which are going to take precedence over attending a seminar. So the next seminar will be, I think, on the 27th or the 28th of August. And I believe it's Blake Myers and Sebastian Belanger.
I think it's the 25th, Robbie.
Is it the 25th?
Yeah.
Okay, the 25th. I think
Blake, yeah.
Blake Myers and Sebastian Belanger from the Donald Danforth Institute in St. Louis. So we look forward to seeing you again then, hopefully, and thank you again for joining. Maybe some links from David Horner, from, who else did I mention that you spoke to? I'm just scooting back through
Nikolai.
these.
Nikolai.
From Tanner, from Nikolai. Yeah, and maybe a discussion as well with some of the other people that are generating genomic resources would be useful. So Craig, thanks very much. Thank you everybody for taking part, and hopefully we'll see you on the 25th of August. Take care.